Welcome student to this tutorial of physiology. So today we are going to do the introduction to physiology. Okay, so this session is going to introduce you to the course of medical physiology. Okay, so if you haven't subscribed to this channel, please hit the subscription button and like the video so that you keep on supporting us. Okay, so introduction to physiology. We must be able to define different terms related to physiology. Okay, so physiology is basically the science of the function of the living system. So this physiology basically looks at how the body functions. In other words, we define physiology to be the mechanism of the body function. Okay, mechanism. How does the body function? So we want to understand how this body functions. Okay, how does the body tell someone to say that right now you are feeling hungry? Okay, how does the body tell someone to say that now you can lift the bucket? Okay, how does the human body knows that now I need to go urinate? And those are basically the mechanism that we are talking about when we are describing the physiology. Okay, so in this introduction, we are basically going to define some few terms. So physiology, maybe we can try to go into describing it. How is it different from anatomy? So anatomy basically look at parts. Okay, you can look at the parts of the, let's say we can look at body parts. Specifically, maybe we go to the liver, then you start describing specific parts of the liver. But when it comes to physiology, we are going to describe how the liver works. Okay, so anatomy is about where, however, the physiology is about how. Okay, how that particular function is carried out. Okay, so this physiology part, we are going to look to take a look into the body fluid compartments, understand various body fluids in compartments. Okay, so within this body fluid compartment, we are going to find terms like the same compartment itself, fluid, osmolarity, equilibrium. So what are compartments? Compartments, we can say subdivided areas where this fluid is collected. How about the fluid? Fluid, you're just looking at the liquid state of a substance. Okay. How about the osmolarity? Osmolarity is about the number of electrolytes contained within a solution in a liter of a solution, to be specific. How about the equilibrium? Equilibrium, we're looking at there is equal distribution of this fluid. Okay, let's now dive into this body fluid compartment and solute. So the body fluid compartment and solute, we must be able to know that we have body fluid in this title, we have compartments and solutes. So the body fluids, we are looking at the liquid state of this particular substance in the body. And the compartments, we are looking at the subdivided areas where this fluid is actually found. And then we are looking at the solutes. The solute, we are looking at the number of or amount of these particular ion substance, okay, like electrolytes, sodium, calcium, potassium, sodium, magnesium, other, okay, are being contained within these body fluids and are located within the compartment. So the human body collectively contain about 18%, 7%, 18% of protein, 7% of mineral, 15% of fat, 60% of water. Okay, this is approximately in someone who is having, who is considered to be young adult weighing 70 kg. We must be able to note that this body fluid can differ depending on the various factors. Okay, so there's a point here where it's saying water percentage can vary from 50 to 70%. And in what ways then this water percent can vary? In case of fat content of the person, the age of the person, as well as yes, for specifically the age of the person matters. The age of the person matters and the fat content. Okay, among others that you can research. Okay, now How have we gotten this body fluids collectively? Is we are trying to get the intermediate, a male who is weighing 70 kg. So in other words, we are saying in a male who is weighing 70 kg, this is the amount of body fluid they should have. 60% of water, 18% of protein, 15% of fat, and 7% of minerals in weight. Okay. So we have two major body fluid compartments in the body. Number one, we have the extracellular body fluid compartment and we have the intracellular body fluid compartment. Now get this. These are just terms that can try to confuse you when they are very simple. Look at the extra and cellular fluid. So we are basically looking at the fluid that is found outside the cells. Okay? And how about the intracellular fluid? It is the fluid that is found inside the cells. Okay? Then among these, between these two major body fluid compartments, we have some varying amounts within them. Okay? So the intracellular fluid is about 2/3, okay? 2/3 of the total body fluid compartments. So we are going, maybe we are going to take the total body water. The total body water that approximated in an adult 70 kg male was 60%. So that is going to be the point of reference. So 60% of this body fluid, okay, is going to be subdivided into two major body fluid compartments. We have the intracellular. Then we are going to say 2/3 times 60. That is going to be 40%. And the extracellular, which is 1/3 times 60, that is going to be what? The 20%. So we have more of intracellular fluid, fluid that is found inside the cell, and the extracellular fluid, fluid that's found outside the cell. Now let's explore more about this intracellular fluid. Remember, let's look at this. This is a cell, okay? A cell, and then within this same cell, there is surrounding fluid outside. So the fluid that is found within this cell is considered to be intracellular fluid, and the fluid that is surrounding the cell is the extracellular fluid. Same here. When we look at this, this is the blood vessel. The blood vessel is having some cells within it. Okay, so the blood vessel is also its own compartment. It is having cells like the white blood cells and the red blood cells as well as the platelets. So fluid that is found in these red blood cells, white blood cells, and platelets, they are considered to be intracellular fluid. And the fluid that is found in outside the cell is considered to be what? Extracellular fluid. Okay, and then we have uh, the fluid I described to say that fluid that is found outside the cell. Okay, now let's assume of a liver cell. A liver is an organ. Within it, it's having cells. Those cells are hepatocytes. Now, the fluid that is within the hepatocyte is going to be considered the intracellular fluid. Why? Because it is within the cells of the liver. How about the fluid that is going to be outside the cells of the liver, but this fluid is within the liver? It is going to be called the interstitial fluid. Okay, so we only have two major fluid compartments. It's either the fluid is inside the cell or it is outside the cell. However, there is something unique about extracellular fluid. Extracellular fluid can be subdivided into three other fluids that we are going to describe. But for now, let's concentrate into these two major compartments and see their differences. Okay? So, and more emphasis on the body fluid. We have the intercellular fluid and the extracellular fluid. Look at this. We have this fluid, which is considered to be intercellular fluid because it is inside the cell. But how about the fluid that is in between these spaces? It is considered to be interstitial fluid. Okay? Which is the component of the extracellular fluid. So I said intercellular fluid is one major on its own. However, the extracellular fluid, it is a component of a number. We have three extracellular fluid depending on where this fluid is located. So the fluid that is located outside the plasma, it is not in the plasma, which is the blood vessel, and it is outside the cell. we call it the interstitial fluid. And then we have that the interstitial fluid is part of the extracellular fluid. What is another extracellular fluid that we know? It is the plasma fluid. This plasma fluid, it is on its own. It's the blood vessel has its own. It's more like it's an own compartment. So it is having the cells and it's having the fluid part. Of course, these cells like blood cells are having fluid in them, and they are going to be considered intracellular fluid, regardless where they are. For as long as fluid is inside the cell, it's considered to be intracellular. However, the extracellular fluid matters where it is located. So since this fluid is located outside these cells, it is going to be considered the plasma, a unique extracellular fluid. However, the interstitial, it is not in the plasma. It is in other cells, outside other cells. It's going to be interstitial. And then the third uh, category of extracellular fluid, we have fluid that is found in the body cavities, like synovial fluid, like the pericardial fluid. We have the cerebrospinal fluid. Those are considered to be transcellular fluid. Let's explore more on this one. So I described to say that extracellular fluid is a general term, but within extracellular fluid, we have some subdivided categories. We have the interstitial fluid and the plasma fluid. What's the difference? I stated that for intracellular fluid, we have no problem. For as long as it is inside the cell, it is considered intracellular. Wherever this fluid is, for as long as it is intercellular. However, we have the problem with the extracellular fluid. Why? Because where this fluid is found matters. If it is outside the blood vessels and outside the cell, it is considered interstitial fluid. For example, fluid that is not inside the cells of the liver is considered interstitial fluid because it is not in the blood vessel. However, the fluid that is outside the cells of the blood vessel is considered to be the plasma fluid. And what is the last one? What is the last subdivision of extracellular fluid? Is the transcellular fluid. And what is it? The transcellular fluid is the fluid that is found in the body cavities, like the pericardial fluid, the cerebrospinal fluid, and the synovial fluid. Those are compartments of body fluids. Okay. Now, we describe to say that we have body fluids, compartments, and solutes. Now, compartments, what separates these body fluid compartments? What separates intracellular fluid from extracellular fluid? What separates them? Okay, so what separates the extracellular fluid and the intracellular fluid is what we call the plasma. So as we can look here, this is the this is an animal cell. Okay, it being an animal cell, it is having the plasma. Okay, the cell membrane here. So the fluid that is inside this animal cell is considered intracellular. However, the fluid that is collected outside is considered the outracellular fluid. If we zoom in the cell membrane, the plasma membrane, that's another name for cell membrane. We will look at to say that this is the barrier, the cell membrane that is in between. As you can see, we've gotten the section from here. Okay, this is the cell membrane that makes a barrier between the intracellular and extracellular fluid. Okay, what separates interstitial fluid and the fluid that is found in blood? Okay, what separates the interstitial fluid from the plasma is this capillary membrane here. This is what separates the interstitial fluid from the plasma fluid. And the last component, which is the transcellular fluid. For transcellular fluid, it is just found in the body cavities. There is no unique about what separates it from other body fluid compartments. In summary, the total body water is 60%. Intercellular fluid is 2/3 of the total, which is 40%. The extracellular fluid is 1/3 of the total body water, which is the 20%. Then the extracellular fluid is further divided into three. We have the interstitial fluid, fluid that is found outside the cells. We have the plasma fluid, fluid that is found in blood, and the transcellular fluid, fluid that is found in the body cavities. Now let's distribute the 20% that we got from the 60% total body water. So the out of the total body water, which is 20% of the extracellular, the interstitial have 3/4. Once we multiply 3/4 times 20% of the extracellular, is going to give us the it's going to give us a certain amount which we are going to calculate. How about the plasma? The plasma is 1 out of 4 of the total 20%. And the last, the transcellular, is too negligible. It's usually not calculated, but regarded to be somewhere 0.5 liters. Okay. Now, this is the transcellular fluid that I was describing. Apart from remember I said the extracellular fluid is a general term. Under extracellular, we care about where this fluid is found. We have the interstitial fluid, the plasma fluid, and the transcellular fluid. Interstitial fluid, we said fluid that's found in the cells or between the cells. Okay, the plasma, we are talking about the fluid that's found in the blood vessel. Okay, and then because it's outside the cells of the blood. And then we have the transcellular, the fluid that is collected in these body cavity that's displayed here. We have the synovial fluid, we have the pericardial fluid, and we have the cerebrospinal fluid. These are considered to be transcellular fluid, and they're about 0.5. Now, let's dive into the calculation of how this fluid is distributed in the body. So the normal body of a human being, we assume approximately we have about 42 liters. Okay, we describe to say that we have two compartments, the extracellular as well as the intracellular. The extracellular fluid compartment, we said it is 20%. 20% of the 42 liters is going to give us 14 liters. How about the intracellular? We said it is 40%. If we multiply 40 by 40%, 40% of 42 liters, we are going to have 28%. The intercellular fluid, we say that we have no problem with intercellular fluid. For as long as the fluid is inside the cell, it's considered to be intracellular. How about the extracellular fluid? For extracellular fluid, we have issues. It depends with where this fluid is. Okay? If it is in blood, it is considered to be plasma. If it is in the blood vessels, it's considered to be plasma. Because blood vessels is considered to be its own compartment. It is having fluid that is inside the cell and outside. Now, the one which is outside is considered to be the plasma. Okay? How about interstitial fluid? Interstitial fluid that is found between cells. Let's look at the liver. Let's assume the cells of the liver, the hepatocyte. What is inside is intracellular. However, the fluid that is remain between these cells is considered to be what? The extracellular. Then the transcellular, we have about 1 liter. How is this 14 liters distributed? This 14 liters is distributed because we have different compartments. So the plasma is 1 over 4. 1 over 4 of the entire 14 liters, and that's going to give us 3 liters. How about the interstitial? It's 3 over 4. That is going to be give us 10 liters. And the transcellular, it is too negligible such that we are unable to calculate. Those are body fluid compartments, and this is a summary of the body fluids compartment. Now, if you want to learn more about what is the various roles of these fluids, you can kindly screenshot these slides that I'm showing so that you pass through. They're actually easy to understand. So we have the interstitial fluid, their roles, we have the plasma, their roles, and we have the transcellular fluid and their roles. But in today's lesson, we are going to be specifically going into describe the general function of these body fluids. If you have time, you can pass through those previous slides that I've moved. However, what are the function of the body fluids in general? Okay, if you want to understand the individual function of this body fluid compartment, you can actually screenshot those previous slides and understand more about or read more about it. So they are actually the medium for transport. This fluid is the medium for transport. They transport a lot of substances. They transport nutrients, waste products. They are also transport hormones, a lot of substances. They are needed for cellular metabolism. Okay, this fluid is very much important for cellular metabolism. Okay, solvent, they act as a solvent for electrolytes and other constituents. Remember, it should be in a fluid state, so they act as a solvent to dissolve other constituents of the body, help maintain body temperature. They also help in digestion, they help in elimination, and they also act as a lubricant. Okay, so away from that, we are going to look into the body solutes. Remember, we said we have body fluids, compartments, and solutes. We've described what body fluids are, and then we've looked at different compartments, and then now we need to look at the solutes. What are solutes? Solutes are actually the amount of electrolytes or ions that are contained in a solution, in a liter of a solution. In other words, those are solutes. The solutes we're talking about, they are sodium, potassium, etc. Now, how about these solutes? Why is it important for us to know? Okay, so the solutes, we are just talking about this body fluid compartment that we described. We have the extracellular and the intracellular now at large. There is distribution of these ions and the cations. Remember the cations are positively charged, the anions are negatively charged. So they are distributed across this body fluid compartment. Okay, the extracellular fluid compartment is having both the cations and anions, as well as the intracellular is having the cations and anions. But we must be able to recognize that there are some ions or solutes that are dominant in the individual compartment. For example, in the extracellular fluid, you are able to see that there are a lot of sodium in the extracellular fluid. Okay, why? Because they have issues crossing the plasma. And there are a lot of potassium in the intracellular fluid. However, if we total the individual cations and anions in this region should be equal to total cations and anions in this region. Whenever we have total, if we try to calculate the amount of potassium chloride and other maybe proteins, we put them together, it gives us about 290 millimoles. And then we also total amount of solutes here, it gives us 290. Then we can say that this cell is at a resting state, meaning that there is no activity that is happening. There is no shift. There is no net movement of what? These ions or solutes across the cell membrane. We're together there. Okay. So in other words, we should be able to have the total amount of cations and anions in the ECF, which is intracellular fluid, should be equal to the total amount of cations and anions in the ECF, which is extracellular fluid. Okay. And we have specific solutes which have been displayed in various compartments. So remember, the ECF and the ICF is separated by the cell membrane. And there are some substances which are more dominant in the ECF, like sodium is more in the extracellular and potassium is more in the intracellular. This is the distribution of all these solutes across the body fluid compartments. We have proteins in the plasma. We have more proteins in the plasma actually, because they also have issues crossing they are large molecules. They are unable to cross the capillary membrane to go into the interstitial fluid. Okay, and we have the intracellular proteins which are found inside the cells. However, once we total the ECF cations and anions and the ICF cations and anions, they should give us a total for us to say that the cells is at at equilibrium. Okay, so outside the cell and inside the cell, we need to have same amount of solutes. However, if there is net movement, like the way we've seen the movement of sodium, the movement of potassium going inside the cell, if there is net movement, then we can no longer call that the cell is no longer at rest at that moment. Okay, so the composition, let's talk about the composition of these body fluids, solutes, which they are the solid distribution across this body fluid compartment. We have the extracellular fluid and we have the intercellular fluid. Extracellular fluid we are talking about is the plasma, okay, and the intracellular fluid, which is separated by the what? Blood capillary, meaning that these solutes are able to diffuse because blood capillary is able to allow the process of diffusion. So sodium is more dominant outside the cell than inside the cell. It's about 142 millimoles. And sodium inside the cell is little, about 10 millimoles. Potassium is little outside the cell. However, it's more inside the cell. The distribution calcium, we have magnesium, we have chloride, we have bicarbonate, we have phosphate, and we have proteins. outside the cell we have about 70, and inside the cell we have about 200. However, if we total the intracellular and the extracellular, all of them combined, we must be able to come up with a osmolarity of somewhere approximately 290 milliosmoles per liter. Okay, milliosmoles per liter. These are the units for osmolarity, meaning that we are just looking at the composition of these solutes within a liter of a what? Of a solution. And the normal osmolarity of blood is in between 280 to 290 milliosmoles. Then the osmolarity of blood is normal. So this is the distribution of solutes across this body fluid compartment. It's actually very much cardinal when we start looking at the neurophysiology, explaining the genesis of membrane potential. They are very much important to refer to these ones. Therefore, it's very much important that you learn about them. Same concept here. It was just a picture to show that it can range in osmolarity from 281 milliosmoles to 290 milliosmoles somewhere. Then it's considered to be normal. In summary, we said body solutes, the amount of electrolytes contained in a liter of a solution, okay, they are distributed across the compartments, the extracellular compartment and cytoplasm. However, they are both distributed. The anions and cations are found in both regions, the intracellular and the extracellular. However, if we total them, they should be able to be equal for us to call that the cell is at rest, and that is it has attained its equilibrium. There should be no net movement of these solutes. Otherwise, if there is a movement of these solutes, then we are going to consider it to say that it's no longer at rest because there is no equilibrium. Okay, and then these were the normal values of various solutes, and then once we total them together, they gave us 290 milliosmoles per liter. Okay, so osmolarity, we are looking at the number of solutes that are contained within this solution. And how much is this solution? It is 1 liter. And that's it about the solutes. So we've covered the body fluid compartments, we've looked at the compartments, we've done the solutes as well, the solute distribution across these fluid compartments, and then now we want to look into measuring body fluids in compartment. We want to measure this fluid. It's very much, it has its own clinical significance. So we must be able to measure this fluid body compartment. How do we measure it? This is how, this is and what, that is what is going to be described under this subheading, which is saying measuring body fluid in compartment. Measuring is just the act of us taking the amount that is available, body fluids in compartments, looking at different compartments of the body specifically. Okay, there is what we call indicator dilution principle. What is indicator dilution principle? Indicator dilution principle is that we have a dye. It is a principle that states that we have specific dyes which can only be contained in particular body compartments. Okay, and these dyes are going to be injected in the human body. Since they have the ability to cross some barriers within these compartments, then we are going to measure the amount that is available. What do I mean? What I mean is, if we have a dye, we want to measure the total body water. We want to measure the total body water. And we said that the total body water is in two compartments. We have the intracellular and the extracellular. Okay. Now, what should this dye be? This dye, as you can see here, it should be that if this is the plasma membrane that we've cut in between, and then we have the ECF, extracellular fluid, and the intracellular fluid. If you want to measure the total body water, then this dye should have an ability to cross the plasma membrane, also known as the cell membrane. If it crosses the cell membrane and then it is contained within this component, then we can be able to tell that we have measured the total body water. So if you want to measure the amount of the entire amount of water that is within this human body, we must inject this person a dye. Dye is just a substance having a color to stain that particular body compartment. And this dye is going to stain the entire body compartment, which is the what? The the intracellular and extracellular. And this dye should have an ability to cross the plasma membrane. So it's very much important. They'll ask you these questions in physiology. They'll ask you about what dye is used to measure the total body water. And what criteria do we use? The criteria is that whenever you're using this dye, it should be able to cross the plasma membrane. That's why I was teaching you about the compartments, what separates the interstitial from the plasma, which is the capillary membrane, and what separates the intracellular from the extracellular, which is the plasma membrane. Now, what if we want to measure the ECF, the extracellular fluid? Remember, we want to just get the extracellular fluid, meaning that since we have this total body weight, total body water, but we are only interested on the extracellular. So meaning that if we are able to inject this person a dye, a specific dye, as you can see here, this dye should not be able to cross the cell membrane because we only want to concentrate on the what? On the extracellular fluid. Once it crosses the cell membrane, it's going into the intracellular, and then we are going to get wrong readings. So this particular dye should only stain the extracellular. And what is the criteria? It should not cross the plasma membrane here for us to measure the ECF. How about the plasma? How do we measure the plasma using a dye? For us to measure the plasma using the dye, we must be able to know that we have one major demarcation, which is the plasma membrane, okay, which is separating the extracellular from the intracellular on the left side and the extracellular on the right. Remember, this is the ECF, and this dye should be within the ECF. Now we want to break down further the ECF, since we said it is subdivided into interstitial having 10 liters and the plasma having 3 liters. Now we've gotten only the ECF and further break it in between. So the one which is shaded in red is the blood vessel. We only want to know the amount of fluid in the blood vessel. And this dye should only stain the blood vessel. It should not cross the capillary membrane. Because if it crosses the capillary membrane, then we are now measuring the ECF, because it's going to contain the entire compartment of the ECF. Do you get that point? So the criterion for the plasma, when you want to measure the plasma, is that this dye should not cross the blood capillary. It should just be within the blood, so that when these machines that we are using, they detect this specific dye in this blood capillary, it's going to calculate the amount of water that is in the blood capillary. And if they find 3 liters, then they'll be able to conclude to say that it is the normal amount that should be there. Remember, we're just dealing with the approximation. Now, we've calculated the total body weight, we've calculated the extracellular fluid, and we've calculated the plasma. How can you calculate the intracellular fluid? Remember now that we have the ECF and the plasma. We can use the formula which says total body water is equal to ECF plus the ICF plus the ECF, which is intracellular fluid that is found inside the cell, and the fluid that's found outside the cell is the what? Is the total body water. Remember, we said that these are two major components. So we can substitute total body water, we put the value that we found, then we put the ECF value that we found, and then we can calculate the ECF like the way it's been expressed here, which is ICF is equal to total body water minus ECF. It's just manipulation of the formula. Same here, if you want to calculate the interstitial fluid, since we already have the plasma here, we can just say the ECF minus for the plasma. Then we are done with this. Now let's look detail. Let's summarize what everything that was happening. So this marker that we are talking about, markers used for measuring body fluid compartment. Markers used for measuring fluid compartment. Remember, this marker we are talking about is the dye. This dye is injected in the human body, and it has specific criteria, and the ones I was describing in the previous video, so that we can measure the body fluid in compartment. Number one, How do we measure the plasma? I said the substance should not cross the blood capillaries, because if it crosses the blood capillaries, it's going into the interstitial fluid, and the interstitial fluid plus the blood capillaries plus the plasma, it is making the ECF. So we are not measuring the extracellular fluid. We want to only concentrate to the plasma, and this fluid should stay within the blood vessel. Therefore, it should not cross the blood capillary. Its ability should not cross the blood capillary. And what indicators are these? You find we have Evans blue dye, we have radioiodinated fibrinogen, we have radioiodinated albumin, which are indicators that are used to measure the blood plasma. So you find this question in MCQ. Which of the following indicators is used to measure the blood plasma? It are these. But the ones which are highlighted in red, they are the most available one or most used one. Evans blue dye is used to measure the plasma. The criterion, it should not cross the blood plasma. Remember, should not cross the blood plasma. How about the ECF? The substance should not cross the the can cross the blood capillaries, because blood capillaries are still part of the ECF, but they should not cross the cell membrane. Remember the one I was talking about here? The demarcation in between here is the cell membrane. So it can cross the blood plasma, the blood capillary, sorry. It can cross the blood capillary, but it should not cross the cell membrane. Because if it crosses the cell membrane, then it goes into the intracellular fluid. Then we are measuring now the total body water. I hope it's clear. And what is the most common one that we use to measure the ECF? The most common one which we use to measure the ECF is inulin or mannitol. Okay, so most of the questions that you find in UNZA papers, they use mannitol. Mannitol is the most common one that they use, but inulin, according to other sources, is the most common one. But the mannitol is the most likely one, so that they try to see if you understood or if you read about others. So we have isotonic solution, sucrose, inulin, mannitol, sodium chloride. They are all used to measure the ECF volume. They injected in the body, and then they are going to create a stain. If it is blue, then the machine is going to measure the fluid that is in blue. That's what we mean. And what's the criteria of the fluid? It can cross the blood capillary because it is part of the extracellular fluid, but it should not cross the cell membrane, because now we are going now to body compartment, which is total body water. I hope that was clear. Then we have the total body water. How about the total body water? Total body water, we have the intracellular and extracellular. So we need this dye to cross the what? The what? It should be distributed within this compartment. Just like the way it's been distributed here, the total body water, it's been distributed across the component, the ECF and the ICF. So it should be able to cross the blood capillaries. But the most ideal criterion is that the substance distribute uniformly in the ECF and the ICF. And what substances are used to measure the total body water? Heavy water, dehydrated water, we have titrated water, we have the aminopyrin, we have the antipyrin. All these are used to measure the total body in water. Okay. Now, how to measure body fluid in compartment? Remember, we have injected this dye, a particular dye in the human body. It is being injected in the human body, the particular dye in the human body. Okay. It is used to measure. We are going to measure using this dye which I've injected. And there are some criterion that we must follow when we're injecting this particular dye, okay, about this dye or about what should happen in the body. So how to measure? We're talking about now the normal calculation. How do we calculate individually? How do we calculate? Okay, the body fluid compartment is using a dye and should meet the following criteria. One, they should mix uniformly across that compartment, throughout that compartment. So if you are measuring in the plasma, you need to make sure that that particular dye is distributed within the plasma only, because we are concentrating in the plasma. Okay, it should be non-toxic. This particular dye should not be dangerous or should not be harmful to the body. Okay, and then it should have no metabolic activity. Metabolic is we mean to say that it should not take place in any activity, the catabolism or anabolism that is happening in the body, because in that way it's going to be used up. We are going to get wrong readings. Okay, it should have no effect of its own in the distribution of water. Okay, it should do nothing actually. It should either it must be unchanged during the experiment, or if it changes, the amount change must be known. So if you put in and then it get to be mixed up and then it get to be used by the body, you must be able to detect that amount that was used by the body, so that you subtract it from the body fluids that you are going to measure. Should be relatively easy to measure. It should be something that is detectable. It should be something that you know the amount exactly. Okay, so the dilution principle. The dilution principle, now we are specifically looking at how everything is done. So the dilution principle uses the principle of mass conservation. The dilution principle we are looking at, remember, we are injecting this dye in a particular human body. This dye is having a specific mass and it is having a specific concentration. And then we want to measure the volume of those body fluid compartment. How are we going to do that? Pay attention and listen to what I'm going to tell you. So the dilution principle uses the principle of mass conservation. What is the mass conservation? We know that mass is never changed. Okay, the mass of a human body of the human body on the earth never changes, not even on the moon. Same with this principle that we are using. The mass conservation is that mass never changes. Okay, so the mass we're talking about is the mass of this particular dye. It's not going to change. If it is 3 grams we inject in the human body, it's going to be that same 3 grams. It is not going to change. In as much as it can be distributed, but still it's going to be 3 grams. However, the concentration will change. Why will concentration change? Yes, the reason is you can't compare the concentration of, let's look at the concentration of sugar. You have this big cup, and then you put one cup of teaspoon. Okay, I mean you put one uh, um, teaspoon of sugar. Okay, you get this teaspoon and then you get this sugar, then you put in this water, and then someone who is going to put uh, 10 teaspoons of sugar. There's going to be different concentration. The concentration we're talking about is in that area of glucose, is how sweet the solution is going to be. You can't compare 1 teaspoon of sugar and the one who has put 10 teaspoons of sugar. So in similar manner, we are describing a dye here. So this dye is having a particular concentration, but once you inject in the human body, it's going to be distributed and it's going to be changed, the concentration. So the concentration will change, however, the mass will be the same. I hope we are moving together. Now, the thing is, we have an X gram of marker, which is a dye. The gram we're talking about is a mass. So, for tracing sake, everything that is in mass, I've written it in red. Everything that is in concentration, I've written it brown. And volume, I've written it blue, so that we can follow each other. So an X gram of marker is injected into the compartment. Remember, this is the dye we've injected in the compartment. We are going to take it as X so that we come up with the formula X, meaning that it could be anything. It could be 10 grams of the dye injected in the compartment, 3, okay, and then measure concentration and equilibrium. We want to measure concentration when what is this equilibrium? When this particular dye has fully distributed across this compartment. Okay, so we want to measure them at equilibrium. So at that point, we can easily calculate the volume using the formula, which is saying concentration is equal to mass over volume. Remember, we know the mass of this particular dye, and we can calculate the final concentration of this particular dye after it has been mixed. Then we can know the volume of that particular compartment by using the formula. Concentration is equal to mass over volume. Then we can make volume, because we're interested in the ECF and the ICF, and those are volumes. They are body fluids compartments and they are volumes. So we can easily calculate volume specifically maybe if we want to be taught to calculate the ECF, we can calculate volume to be mass of that particular dye over the concentration. The concentration we're talking about is the final concentration of that particular dye now that it has attained its equilibrium. And what is this equilibrium? Is when it has been fully distributed across this compartment. Okay, so this is the principle to say that indicator mass A is equal to volume A times concentration. So mass is equal to volume times concentration. We are talking about the indicator of a dye that we're injecting in this liquid. Okay, indicator mass A. Then we have this beaker here. It's having this liquid and it's having this liquid. Now, the principle of mass conservation, we say that mass is never changes. Okay, unless otherwise, but it never changes. The mass never changes. Let's take it. The mass never changes. Then we have, for us to calculate the mass or volume, we need to know the concentration of that particular dye and we need to know the volume. So if we know the mass of this particular dye and we know the concentration of this particular dye, then we can calculate the volume of that particular compartment. How? This is how. The indicator A, we've put here the mass A, if in as much as we mix in the beaker, but it's going to equal the mass B. Okay, why is it equal the mass B? In as much as we have put it in this beaker, but still the mass does not change. So the mass of this indicator still remains the same even if it is evenly distributed. As you can see, these particles are distributed. But we are saying that if we incorporate these parts and put them together, they are going to give us that same mass. Let's assume if the mass was 3 kg, 3 grams, this 3 gram, we are going to get it at the end of the experiment. Okay, so indicator mass B is equal to volume B times concentration B. Now, remember this is the volume that we had to put this indicator. Now, let's assume that this volume is the body fluid. Let's say it's the ECF, extracellular fluid, that we've injected this dye. How do we come up to calculate the volume of B? Is that we know that indicator B is equal to, now remember we said indicator mass A is equal to volume A, concentration A. Here, since we said that the mass never changes, we can say that the volume B, because we know the volume for this particular solution, which is B, times concentration B. Concentration B is that now that we have mixed the dye and the volume that is in here, we can calculate the final concentration. In other simpler way is, at this moment, if we know the mass of a dye, and then we know the concentration at the end, we can calculate the volume of that particular fluid that we injected this particular dye. Yes, it is as simple as that. Now let's look at the practical example how to do it. The calculation. A 115 milligrams of sucrose is injected into a 70 kg man. Remember, this is the mass of sucrose. Sucrose is a dye that we've injected into a 70 kg man. 70 kg here is just a distractor and it plays no role. Okay, we are interested in mass, volume, and concentration. The plasma sucrose level after mixing is 0.01 milligrams per milliliter. Remember, after mixing, plasma sucrose level 0.01 milligram per milliliter. This is now the final concentration that we will have. Okay, we have now this final concentration. Let me break down this concept. We have a dye of known concentration, but after mixing with a solution, then we have the concentration that is going to be here. Remember, I said we are utilizing the principle of conservation of mass, meaning that the mass is not going to change at this particular moment. It's going to be like this. Nothing is going to change here. So the mass is like this. If 5 milligram has been metabolized, remember the criterion, we said that the dye should be non-toxic, should not play any part, should not be metabolized. So if 5 milligrams has been metabolized during this period, then what is the ECF? Remember the ECF, we are looking at the fluid compartment of this particular body. Okay, it is the body fluid compartment. So I want to calculate the ECF. This has been injected in an ECF, and we know that sucrose is the one which is a dye used to measure the ECF. Okay, So how are we going to do this? We are going to use the formula. Volume is equal to amount of the indicator. Amount of the indicator, we know it is the mass, the amount of a substance, the amount contained in the substance. That's how we describe mass. So the amount of indicator over final concentration in the ECF. We had to inject this particular dye into the ECF, which is the body fluid compartment, and we need to calculate the final concentration. We are now combining the ECF and the dye so that we can come up with the final concentration. Now, remember, we want the volume. The amount of the indicator is what? Tell me the amount of indicator. The mass, right? And we said it is 115 that was injected in this man, but 5 milligrams was what? Injected, meaning that we are going to subtract the 5 milligrams from 115 because these 5 milligrams who cannot be miscalculated. It has been used in the body, and we need to subtract it. If we ignore it, we get wrong results. And the final concentration is this after mixing the dye and the ECF. Now let's look at it. This is just a formula, the layout to say how we calculate it from the principle of conservation of mass. We've broken down into this far. Now let's do step-by-step calculation on how we're going to calculate the ECF. The ECF is the volume in simple terms. So step 1, we are going to calculate the effective amount of sucrose in the ECF. Remember, effective amount of sucrose that was injected. We are going to have, we had 115 which was injected. However, 5 was metabolized. So we are going to have amount of 110 milligrams of mass, which was not metabolized, which stayed, which never changed. It was the one which should come up. The 5 milligrams were subtracted because it was metabolized, and the criterion, remember, you should know those criterion by heart. It should never be toxic. It should never metabolize. Okay, it should never have any distribution of water. Okay, it should never be unchanged, and if at all it get to be unchanged, we must know specific amount. And since here it was changed, we know the specific amount which was changed, 5 milligram. We're going to subtract 115 minus 5 milligram. That is 110. We have the mass and it never changes. Let's go now. I try to detect the concentration. The concentration after adding, we said that after adding the indicator and the ECF, it gave us 0.01 milligrams per milliliter. So this was the final concentration of the entire dye and the what? And the ECF. Okay. Then we want to calculate volume. We can use the formula now. Volume is equal to mass over concentration. What is the mass? The mass is 110 milligram. We are trying to divide it with its concentration, which is 0.01 milligrams per milliliter. So we'll cancel the gram, and the gram will cancel. We'll remain with milliliter. So if we punch in these values into your calculator, you find that we have 11,000 milliliters. And this 11,000, for us to get, in case they ask you to state this value in liters, you can easily divide it by 1,000 and then you convert it to 11 liters. This is all about the EAST introduction. I hope you liked the video. Like always, if you enjoyed the video, please subscribe and like the video. Thank you very much.